Okay, great. Welcome everybody
Welcome
to.
back to Introduction to Genetics and Evolution.
I'm sure you've heard that before.
Yeah.
Many a times.
Um, so I think we'll just start by having everybody introduce themselves. Um, Mohammed, just how you got interested, maybe in the course, your name, and maybe where you're from,
Yeah.
and then
Yeah.
maybe what you got interested.
Fun fact. We start with Abby.
Hi, yeah, so my name's Abby. I'm from Vancouver in Canada, and I was just interested in the topic as something that I really enjoyed in school and wanted to revisit now. Yeah, and yeah,
What do you do now?
I work in financial services, so something completely unrelated.
Okay. That's excellent. What have you liked, or what have you liked the most from the material? What piece of genetics or evolution have you found especially interesting?
I've most enjoyed the more biochemical stuff, the some really sort of small scale things, but that's more tied into.
I think we froze a little
A
bit
little
there.
bit. Should we move on to Elaine?
Yeah, sure. So I'm Alain. I'm from France, Paris area, and how I got interested in that, it's kind of personal, but I can tell it. I am the father of a son who has called a syndrome called DUP15Q, which is duplication of chromosome 15. And what happens when at one time a genetician told us they would do a genetic analysis. So she did that, which took a long time, like 6 months, and she gave us a very complex report in which I couldn't understand a word. So I said, I need to do something about that. And I found out this Coursera course and got and subscribed to it. And I must say, before we go on too far, really thank you, Professor Noor, and also Blair. It's really awesome what you're doing for free. I mean, it's just, I'm really impressed. I'm telling all my friends around that I have to go check Coursera and really doing a lot of ads for you guys.
I'm glad you enjoyed it. Thank you very much. Has it been helpful in understanding, the background for how that condition came about? A bit of a background thing going
Yeah,
on.
there's background.
I think Gregory needs to mute his microphone and use headphones.
Yeah. There we go. That's better. Can you hear us now again? Eileen, can you hear us?
Yes, sure.
Okay, great. I was going to say, did the material at the beginning of the class help you understand your son's condition a little bit more about how it came about and things?
Yes, yes. I still have, tons of questions, but I don't want that to become a private genetic,
No, I understand.
But yeah, it helped a lot. It really helped a lot. I understand better. And you know what? I'm going to go back to see that genetician in Paris hospital. I told her already I'll be back in the spring with more questions. I guess she'll be impressed by what I know.
That's excellent. Cool. Well, it's a pleasure to meet you. We move on to James.
Hi, I'm James, and I was born in Cleveland, Ohio, but I actually live in Taipei, Taiwan now.
Oh, neat.
And I'm actually adopted, and so in 2009 I started doing some direct-to-consumer genetic testing, and really got interested. One of the things that I found out was I have a genetic disease called hemochromatosis, which causes too much blood in the iron. And one of the other things that I got interested in is mitochondrial DNA. And so I actually have a program for analyzing mitochondrial DNA test results from direct-to-consumer tests and determining what the best haplogroup map best haplogroup match is for your test results.
That's very cool. You know, mitochondrial DNA, the mutation rate for it is much, much higher than for most nuclear genes, and that's part of why it actually gives such a clear phylogeny and things like that. So,
right. It's really exciting to be able to see, like exactly when humans first got out of Africa, and when humans migrated to North and South America. Just being able to see that from just this one very small part of the genome.
Oh, yeah.
Really exciting.
Is it 16,000 bases or something like
Yeah.
that, right? Very
Yeah,
small. Much bigger.
16,000.
Plants have
Yeah,
big.
16,569 is on average, but
Okay.
very.
Excellent. Well, it's a pleasure to meet you. We move on. Who's next on the list there? Raheel.
Hi. So I'm from Pittsburgh, Pennsylvania, and I work at the for University of Pittsburgh as bioinformatics analyst, and I deal with lots of next generation sequencing data, which is humongous amount of data, and it takes so much of time to digest the data and then make it easier for the professor to understand. So I have to bring everyone on page as well. So what I'm doing. So yeah, it's kind of a challenging task, but it's other than I used to work on microarray before, but microarray will give you limited information of what the gene is behaving, which gene is expressing, but when you look at the next generation sequencing data, you see how the transcripts or the isoforms within the gene are behaving. They can behave in different way, and then do those pathway analysis. It's very exciting, and but my main interest in biology was.
The video seems to have frozen.
Raheel, yep, froze a little bit.
It froze right after you said your main interest in biology was. Yep.
Well,
we'll come back.
Yep.
Let's do that. So let's go to Shuhong and come back to Raheel in a minute.
Hi, I'm Shung Bishash. I'm a fifth year medical student in Chittagong Medical. I'm from Bangladesh. I'm very much interested in pharmacogenomics and targeted therapies and prenatal diagnosis. I came to this course to know more about the genetics and its relation with evolution processes. And Professor Noor was very much comfortable with it. And now I believe that natural selection is a reality. Before, in our country, it was not totally taught well. Bangladesh is not concerned with natural selection and genetics. But now, after doing this course, I know that natural selection is a mathematical inevitability.
Excellent. You used the right words, too. That's excellent. I'm so glad you enjoyed that material. That's great. Is that the part that you like the most, is the stuff about natural selection?
Natural selection, the genetical drift, and the natural selection is not the same, but natural selection is much more predictable than the genetical drift. And we can predict our future, of what will happen in the future, what will be the evolution of the human and other animals. I think it is possible to predict, but for now it is much more huge data, and we cannot digest easily that, and we cannot predict. Actually, it is all mathematical probability, I think it. And after some more innovations, we will be able to much more sure about our future, I think it.
That's great.
Thank you, Professor Noor.
Thank you. Thank you for joining us. Pleasure to meet you. You want to say something about your background, Ben?
Sure. Well, I'm a PhD student in the genetics and genomics background or program, excuse me, and so I've been at Duke for a little while. I worked here as a technician for a couple of years before I started graduate school, as well. And I guess I got first interested way back in middle school, I guess, just in general biology. And then during undergraduate, I took molecular genetics, and it kind of hooked me from there. So I've been at it ever since. My research here revolves around looking at different genetic mutations that we find in human patients, and we try and interpret them functionally using different model systems. So the main one I use is zebrafish, and so we go in and we can interrogate the genes there and find correlations between the functions of the zebrafish and what it might be doing to cause risk for certain diseases. I think that's it.
Oh, that's cool. Which material do you like the best?
I really like all the mapping. I think all the genome-wide association studies and the next generation sequencing, that kind of finding risk alleles and mapping them and figuring out specifically where they are. And then with my research, I'm also interested in what they do.
Nice. I'm Mohammed Noor. I actually first got excited about biology back also in middle school. In fact, I used to say I really liked it. But then I had a high school teacher which almost pushed it out of me. But fortunately, I persevered in college. Then I took biology and I liked it pretty well. I took psychology and I liked that as well. But it wasn't until I took genetics that I thought, this is great. So actually, at the time, this is a funny story. At the time, actually, I talked to the person that I was supposed to go over my courses with, and there were two other classes offered it. This was at the College of William and Mary. There were two other classes offered that genetics in name. One was molecular genetics, which most people took as senior, and one was called evolutionary genetics. I asked the person who taught, or I asked the person, my advisor, not the person who taught it, if I should go ahead and take evolutionary genetics. He kind of looked at my grades and looked at me and said, kind of a tough class. Maybe you should take something more consistent with your ability. So, needless to say, I signed up for that class immediately. Maybe he did it on purpose. Maybe he wanted to spur me to actually do it. And I loved the class. Absolutely loved it. And that had a lot of the same material. In fact, some of the material in the class you guys took was modeled on my undergraduate class. But I really, really enjoyed it. I'm still very much in touch with the professor who taught that class. He was fantastic. Absolutely captivating. Yeah, so now in terms of what I do here, I mean, obviously I teach classes in genetics and evolution. I can talk later on if you want about the on-campus class a little bit, but also my lab does research in it. For the longest time, I guess, our lab's been really interested in the process of speciation, what makes a new species, and that's why we have that section in the Week 9 videos on speciation in there. And I think actually in those videos we talked a little bit about some of my graduate research specifically on that, but we're very interested in what are the genetic changes it takes to make a new species, what keeps species separate both evolutionarily and genetically, what makes it so, this kind of seagull can't exchange genes with that kind of seagull, for example. But we do it using Drosophila species, using fruit flies. Have I heard about the growler bear? No, I did not know. Is that like a grizzly polar bear hybrid? I have heard of grizzly polar bear hybrids. I didn't know they were called growler bears.
Right. With global warming, the habitat of the grizzly bear has shifted north, where it's overlapping now with the polar bear, and they can mate, and they are creating this hybrid called the growler bear.
Oh, neat. I'd heard that they made hybrids. I didn't know it was called the growler bear. That's cool.
Yeah.
Gregory, we kind of skipped over you because I think you got disconnected. Do you want to?
Yeah, this is my first time to use this, and it was kind of confusing to me, but I think I'm on now, obviously. My name is Greg Lacko. I live in Houston, Texas. I have a passion for conservation biology. I'm also into animal rights. I want to write a book. I'm still working on that. I've enjoyed the course immensely. I've taken numerous courses on both Coursera and EDX, and this one, Dr. Noor, you made us work, man. I felt like I was back in college. I mean, so many of these classes, it's pretty easy to pass, but I've enjoyed the rigor of the class.
Well, thank you. I'm glad you liked it.
Yeah, I really did.
Should we go back to Raheel since we lost him there?
Oh, sorry about that.
No, don't worry about it. So you said the thing that you most enjoyed, and then right then it cut out. It left us all on the edge of our seats.
So I don't know how much you heard from what I said, but I mean, so I think I started talking about my interest in evolution, and did you hear me talking about speciation? And
I don't remember speciation.
Okay, so the reason so it was the concept of speciation was introduced in my 12th grade, and from there onward it started making sense to me what's the main reason why we see so much of complexity among the living organisms on Earth is because of the fact that there is a speciation happening, and the reason for that is a reproductive barrier, and it started making sense that if the other set of organisms are not reproducing with one set of organisms, so they will not be able to pass on their genes to the next generation in one organism. If suppose there are two organisms, organism A and B, and organism A will be able to pass on their genes to their offsprings in the next generation, but not organism B because they belong to a different species. And the reason for that to happen is because there is a reproductive barrier, and that has led to accumulation of so much of complexity that we see around us, and that made me fascinated towards understanding species. That I want to do my PhD further in evolution, focusing on speciation.
Excellent, excellent. I know I have a lot of friends who work on that, so, so if you want some recommendations of labs, I can suggest some for you.
Yeah, thank you.
So you guys, we can take a couple of questions from you guys, and we also printed out some of the questions that people had written either in the forums or that you guys may have written as well too. Is there anything that any urgent issues that you guys want to discuss, with everybody, not necessarily just with me, but with all of you guys?
Mm.
23andMe. Yeah. What about it? 23andMe?
Oh, you talked in the beginning of the class how we should wait before we send these people money and the couple fit. Okay, is it time to do that now? And I wanted to add, I'm adopted. I don't have any knowledge of my natural parents, and I think in my case this will be especially interesting, and I intend to do it. But you haven't mentioned that again, so I thought I'd prod you on that.
That's a great point. You're right, because I did say to hold off on it, but I never did come back and say now it's okay. No, basically what I wanted you to do is to wait until we finished all the genetic mapping things. So basically, once we got to test 1, I think people had most of the background we wanted to get to. There's a lot of issues with respect to that. So first, it's not. I mean, some people will get it and they see it very much as like, oh, I have 4 times greater risk for this disease. I need to go act on it. From the vast majority of diseases that are covered by services like 23andMe, the risks are very statistical. So, for example, you might have had a 0.1% chance of getting a disease. And so, yes, you might have 4 times greater risk, but now it goes from 0.1% chance of getting disease to 0.4% chance of getting disease. It's still, for a lot of diseases, it's still really, really, really remote. Now, there are some exceptions to that, like the BRCA mutations, for example, with respect to breast cancer, the ApoE. I can never remember the number.
E4.
Yeah, there's E4. Yeah, for Alzheimer's. Mm-hmm. Some of those are very large risk factors. If you have that, then the risk is actually fairly high. The biggest thing is
23andMe now giving information on diseases. You had mentioned how 23andMe for a while had stopped doing that.
Yes, so they're now beginning to phase that back in. They've just started to rephase that back in. Now, what you can do, for example, is you can get your genotype from a company like 23andMe, and then there's third-party websites where you can just download all the data from 23andMe, upload it to this third-party website, and it gives you everything 23andMe used to give. So that's sort of a workaround for it. But there's 2 big things to think about. Or actually, I guess sort of 3 big things to think about. One is the statistical aspects I mentioned before. Second, if you do have a family history and if you think there's some risk that you may actually have a major risk factor for disease, it's very important to talk to a genetic counselor. Don't just go in there and try to work it out on your own and go to I mean, most physicians will try to help you too, but their experience is not such that they're ready to help people with that kind of information because they may not be used to that kind of information, especially if they were trained quite some time ago too. The third thing is, there are potential privacy issues with these things too. So there is this, within the United States, there's this Genetic Information Non-Discrimination Act, which prevents, insurance companies from being able to hike up their rates in response to your profile, things like that. However, there's no guarantee that that law is not going to go away. Right? I mean, it exists right now. It did pass almost unanimously. Ron Paul voted against it, but everybody else was in favor of it. But it's possible at some future time somebody will say, no, we're going to get rid of that act. Also, it doesn't cover everything. So, for example, long-term care insurance, so the insurance that you get with respect to, say, for example, if you have a major illness and you need care for a year on it, that is not actually covered by GINA. So, in principle, I've never heard of this happening. In principle, a company with having that information could decide that they no longer want to cover you or need to raise your rates dramatically and things like that. So, there are many issues like that to consider in addition to sort of the genetic facets and the statistical facets. But those are the pieces of advice I'd say that, number one. Well, I guess the fourth too is if you know there's a family history, honestly, based on most studies, it's way more predictive than your genotype.
Yeah.
And everybody.
Well, as I mentioned, I'm adopted, so I don't have any information.
Exactly. In your case, that's
my natural parents. So, I go into a doctor's office, and on the forms they'll ask about family history. Well, I don't have anything to put down there, so maybe this will give me some insight on that.
Absolutely, absolutely. It's certainly better than nothing, that's for sure.
Yeah, James, did you want to speak more about that?
Sure. For adoption issues, there's three main companies, 23andMe that was mentioned. There's also Family Tree DNA and also Ancestry.com, which are the main three testing companies for autosomal DNA. And really, it's kind of hit or miss whether this is successful for adoption searches. If you can get a second cousin match or better, and they're willing to connect and communicate with you, then you're almost certainly going to be able to solve the adoption case. But most people, they just get third cousin or more distant. And third cousin is just kind of a prediction, so it could be fourth cousin, it could be fifth cousin, it could even be 10th cousin. So those kind of matches don't often lead to solving the case. You should also not ignore, there's search angels, and you can get in contact with me later and I'll help you out with this, that can help you with the traditional record search, depending on where you were born.
I've already done that. It led nowhere.
Okay.
My sister was able to find her parents.
Yeah, I was able to do it actually after several years of working with DNA. I solved it through record searches for my own case. But like I said, some people, they have great success with DNA, but it's kind of hit or miss.
Yeah, actually, my 23andMe profile hit with somebody who then we traded family trees with and figured out that indeed I had never met her, never heard of her, and she was a third cousin once removed. Oh, so there's a real connection there. That was great. That was a good topic to bring up. Thanks for mentioning that. I hadn't come back to that yet. Other thoughts from you guys? I mean, otherwise we can start these questions.
I have a question regarding this. So 23andMe, as we know that they do the population-wide analyses, and they look for and they specifically focus on those alleles that are in how they are fluctuating in the population, or they give you the information based on that. So do they focused on are they thinking of focusing on the family, like if I can provide for the whole family and see if they can just focus on that and look for rare variants in the family?
I'm not positive. I don't think they do that. No, what they will do is they will, for example, so I got my genotype and I made both my parents do it. What they will do is they will give you your phase if you provide all that information. So that can help a little bit with that sort of thing. I don't know that they'll do a sub-study within just an individual family. I've not heard of that at least.
I haven't heard anything.
Yeah.
And also, I don't understand when they say you are 5% or some percent Neanderthal. I know that even with the chimpanzee, the difference between the genome is around 1%.
Yeah.
So is it
this question comes up all the time. This is a very good question. So a lot of people ask about this. Now, the difference there is whether you're looking at the sites that are variable or if you're looking at all the sequence. And it's true that if you were to just compare any of us to a Neanderthal, just using every base in the genome, we would be way more than 99% Neanderthal. But we'd also be like 50% Drosophila or something. I mean, that's an exaggeration, but it would be something fairly large on that.
Zebrafish.
Yeah, exactly. 70% zebrafish. All these kinds of things. So it's not using that. What is inferred to have happened is that on the order of about 60,000 years ago, there was some interbreeding that happened with I don't want to say chimpanzees, with Neanderthal, not with chimpanzees. There's some interbreeding that happened with Neanderthal. At that point, a segment of the genome, or multiple segments of the genome, came from the Neanderthal population to the human population. Big stretches of those segments would have been already identical to the human population, but that's what that 1% is including. It's including segments that are identical, but that they came nonetheless from the Neanderthal population. So it's a little tricky there because the percentages don't match up the way one would think. So in fact, I mean, your percent sequence similarity to a Neanderthal will probably be like 99.7, whereas somebody else who had higher would be 99.72 or something like that.
And they are able to identify those segments based on looking at those unique segments that do not match with the human animal.
Exactly. Exactly. So the good thing is, for the last several years, we have had not just one, but even multiple Neanderthal sequences. So with that, we can see that this is something that has come from Neanderthal, and you see a haplotype block that is not broken up. So a whole stretch of single nucleotides that all match Neanderthal all together. It's pretty cool, isn't it? Middle Easterners are very low Neanderthal, so I don't have that much. Other thoughts or questions? It looks like we lost Abby. Yeah.
On the subject of Neanderthals, there's some people that are questioning whether Neanderthals and humans are actually different species. What are your thoughts on that?
That's a great question. So as a colleague of mine once said, and this is a really good quote, speciation is a process, it's not a point. Right? And I think this is the kind of thing that we see with respect to Neanderthal. By virtue of the data that we were just talking about, clearly Neanderthals and humans could interbreed. And some of the offspring clearly were fertile. Because if they were completely infertile, then it would either be 50% Neanderthal or 0% Neanderthal. But we were obviously neither. So I mean, on a continuum, yes, basically we're probably in that very broad gray area between absolutely completely diagnostic species versus absolutely completely not diagnostic species. In terms of where to draw that line, this is something that people who work in speciation actually get very frustrated about. Everybody has their own favorite species concept, and you saw I introduced the biological species concept, which is probably one of the most popular, but there are a lot of other people out there who really don't like it. They say that's an arbitrary line, that maybe we should be drawing the line over here or drawing the line over there. And I think that's where a lot of the debate comes from, is exactly when you start calling it a different species. But ultimately it is, again, it's that whole aspect of being a process rather than a point. It's not that, boom, that one more nucleotide changed and now we're another species. The one exception to that is polyploidy, of course. But we didn't talk about that in the class.
Johan, did you want to say something?
Yes, I heard many times that whole genome sequencing is going to be a diagnostic tool when the cost is lowering down. Huh? But whole genome, in case of imaging technologies, we can see there is this, so the diagnosis is this. But in case of genetics, it cannot say that this is the cause of the disease.
Exactly.
But it can predict that it is the possibility, 5% possibility or 10% possibility. So how do you think this whole genome sequence is going to help us, the clinicians or the doctors, in the future times?
That's a great question.
In case of treatment procedures, it has also very much impact. Like in case of breast cancer, if there is a HER2/neu or BRCA1/2, then the treatment plan changes. But in case of diagnosis, how it can confirm this diagnosis?
Honestly, there's been several times when by itself that it probably can't. So there's when we talked back, I think it was week 3 or something like that, about some of the complications with genetic mapping. And so your point, I should say ahead of time, your point is exactly right. A lot of people think, oh, we have all the genome sequences, we're done. But again, it's not like everything comes labeled and it's all set up. So there's two facets that are complicated. And one would think that we should be able to just map every trait and then really be done. But number one, there's very strong, for many traits, there's strong genotype by environment interactions, right? That by having a particular genotype, you are more likely to have this disease, especially in this kind of environment as opposed to that kind of environment. So with just the genetic information alone, you can't get it. However, with the genetic information, you can say that, yes, you are at greater risk for it, especially avoid this sort of environment. So for example, I'll give a fictitious example, but I think it would illustrate the point. If you knew that you had some predispositions for lung cancer, it would be a really, really bad idea for you to decide to start smoking. So actually pretty much a bad idea to start smoking anytime, but especially if you knew you had a predisposition for lung cancer, that would be a clear indication that you shouldn't do that, a genetic predisposition. The other thing which is potentially achievable, but it will take a very long time and complicated, is epistasis. There's probably a lot of epistasis associated with a lot of diseases. Now, these single marker associations, kind of like the kind we were doing in the class, it's going to pick up very weak signals there, but it's not going to really tell you, oh, based on having an A or being homozygous for A at this nucleotide that you're going to get the disease. However, if you know you're homozygous at A at this nucleotide, and you're heterozygous at this one, and you're homozygous at this one over there, maybe then the predictive power will go up quite a bit. Now that's one of those final frontier things. It's going to be very hard to actually get that in there. And on top of that, we do still have the interactions with the environment too. But I think iteratively we will get a lot closer where maybe not you can just look at the full genotype and say you're going to have this disease or not, but you can then put much greater odds based on the full genotype as opposed to these single marker associations. Does that answer your question?
Yes. Is there any possibility to repair our somatic genes in future?
That's a good question. That's a good question. I mean, what probably would be easier to do than actually, I mean, it's very hard to repair somatic genes because every cell in your body has it, right? So it's very hard to repair it unless you're a one-cell embryo. What's probably more likely is to know that the cause of a particular disease is that you're failing to produce X protein or producing too much of Y protein, and then putting something that will be an antagonist or something that produces more of that protein. That's something that's probably more likely to work than actually trying to change the genetic code of your whole body. But I wouldn't say I'd rule it out. I mean, you never know. Maybe there will be these sort of targeted gene therapy things for, especially for a disease that is only expressed in a small subset of the cells of your body.
Yeah, I mean, one thing, a paper actually just came out where they did what Mohammed talked about, and they used this genome editing technology. It's called CRISPR, but what they actually did, instead of repairing the genome, they introduced genes to help with HIV infections. So they actually introduced genes to elicit a more proactive immune response in these patients to combat HIV infection. So it's just a way that using sort of gene repair technology to introduce, like Mohammed said, like correct proteins or proteins to elicit some sort of positive response in patients. Rahil, did you have something to add?
So, based on my background here, so, I deal a lot, focus a lot on the genomics area, and we tend to leave behind the proteomics area. And I mean, I wanted to continue, I wanted to add in an answer to the question of Biswas, I'm sorry if I can pronounce, I'm pronouncing it correctly, is that we can have more confirmation on how these genes are behaving, like whether these genes will lead to some disease or not, is by focusing a lot on proteins, because this is the end result of the central dogma from DNA to RNA to protein. And at present, we have more number of genes, around 30,000 in human, but we have only few set of proteins. We don't know the whole set of proteins as yet, and how proteins interact with each other, because there is a lot of post-translational modification is going on at the end, at the protein end, and it's the protein that's doing all those functions. And moreover, we are also focusing a lot on small RNAs, or those RNAs that came from the genes that do not form the proteins, and they play a lot of role, which we used to think that those were pseudogenes, they had no role, but now we are looking in these areas. They are controlling, because they are controlling so many protein coding genes in their expression level. So I think focusing on these areas will also result in more confidence, higher confidence in what the certain genes are, how those protein coding genes are behaving.
Okay. Thanks.
Elaine, did you have a question you want to bring up or ask?
I guess my main question would be, is there a follow-up course after this one?
That's a great question. I don't have any others in Coursera myself. However, there are other classes in Coursera if you're interested in the topics. So, for example, well, actually, Rosie Redfield teaches a class called Useful Genetics. It has been offered in Coursera, but she recently communicated with me that she's actually moving it to edX. So that's something to look for there. And it's much more heavily on sort of the personal genotype aspects. It doesn't really have the evolution part. But it seems like that would be a good follow-up class after having this one. Another one, there's a computational molecular evolution class, if that's a topic that's of interest to you. And that one is it's very high level. I'm sure Raheel would have no problem with it. But it's very high level on the bioinformatics and things like that too. But it is offered in Coursera. That would be another one. if you're interested in animal behavior, so the things from, for example, the beginning of week 9, there is an animal behavior class by a group from Australia that's excellent. I actually watched all the videos for that too, and I really enjoyed seeing those as well. So those would be a couple of examples, but I'm not personally offering another one at this time. It took forever to make those videos. I might do someone in the future, but not anytime in the very near future.
Well, I guess, yeah, Gregory was asking just how does genetic knowledge benefit a conservation biologist? Did you want to talk more about that, Gregory, or?
Well, I have a passion for conservation biology. I don't know what I'm ever going to do with that passion, but I used the example earlier on of the growler bear. It's widely known now that with global warming, the habitats are shifting towards the poles, and species are encountering each other that previously never encountered each other before. There's going to be all kinds of fallout from that, but maybe, you know, this interbreeding, hybrids like the growler bear, that's one example. How is this going to affect speciation, extinction rates, so on? How does genetics play into this?
There's a lot of potential directions you could go with that broader question. One of the most common uses for genetics and conservation is identifying taxonomic groups that are not very clear. So, for example, let's say you were studying populations of some sort of beetle, right? And you see, oh, I see this beetle is present in Florida. I see this beetle is present in Louisiana. I see this beetle is present in Texas. Is it the same beetle in all places or not? Right? And it's important from a conservation standpoint to actually know, are these taxonomically identifiable groups or not? And that's something you can with a lot of genetic data say, oh, look, these are diagnostically different from those, therefore they're independently worthy of conservation because, you know, yes, there's cases too where they may just look different, but it may be purely an environmental effect. You know, that maybe by eating the plants they eat in Louisiana, they tend to turn more green or something. I'm making it for the example here. But something along these lines of identifying these taxonomic groups is one of the most intense things that people do. Some people will look at levels of diversity and try to see if, you know, by how much, so pi was one of the measures of diversity we talked about there. People will use these measures of diversity to estimate the population size in a particular area and see, you know, is this a big population or small? Because, you know, for something large like elephants, you can just go count them. But again, going back to this example of the beetles, you know, you see some, you can catch them, but maybe you're catching every single one that's there, or maybe you're catching 1 billionth of the population. So again,
they go by their phylogeny. I mean, maybe not so much genetic analysis.
Both. You need both genetics and phylogenies to it. I mean, all of that would play in there.
We'll maybe have time for like one more question. Does anybody have anything else they'd like to bring up?
Or I could talk a little bit about the on-campus class if you guys are curious too.
Can I bring in one question? If that's okay. So regarding this, you know, online courses, so I want to ask, so if I, you know, how will I get, you know, with other regular courses I take in the class, and I can get references afterwards after I complete the courses because the professor knows me really well. I've been attending his classes. What about the online courses? How can I, once I finish the course, how will I get the references, and that will help me, you know, either pursue my PhD or, you know, or other things like getting a job or.
That's a great question. That's going to vary quite a bit by the kind of online class. For these massive open online classes, I mean, there really is no reference because, I mean, really nobody knows. I mean, it's just a user ID that we see on the bar. What you can get, of course, is, as you probably heard, is that there's the signature track authentication. So that's something that you can get to just show that you satisfied the requirements associated with the class. And the good thing about signature track is it does actually, what you do is you type in like a sentence or something like that each time you do an assessment. And it confirms it's the same person at the keyboard each time who does it. And you also have to show an ID, like a passport or a driver's license or something like that. So then it authenticates at that point that this is the person who actually was at the keyboard. So that's the kind of thing you can do, but you can't get like a personal recommendation, unfortunately, for these kinds of things because, I mean, it was just a lot of multiple choice tests that you did remotely that nobody really interacted directly with you about. Actually, on that note, oh, go ahead.
No, I was going to see if you wanted to talk about the differences between, like,
yeah, on campus and online. Sure. So and this was one of the questions that somebody had submitted as well too. Is what happens with the on-campus class? So the lectures you see are identical to the ones that my on-campus students do. What I don't do is I don't go up there on campus and just deliver that exact same lecture, because that would be silly. They could just watch it online, so it doesn't really make any sense. And this is the idea behind what people refer to as the flipped class. I'm sure many of you have heard that expression before. The idea is that the most valuable interaction you can have with a professor is not the professor standing up on a stage and talking at the audience. It's much more valuable to have the professor directly interacting with you. So what happens is that people in the on-campus class will watch the videos in Coursera, just like you guys do. And then what I do is I give them a brief quiz each day that they have to take, but then gives me a little bit of feedback of what were the problem areas and what were the areas that everybody seemed to get. I spend the first couple of minutes of class on campus describing, or not describing, sorry, elaborating on the points that people identified as confusing. So for a lot of people said, I didn't really understand what you meant about epistasis even after your explanation. I tried to elaborate a little bit further on it. The bulk of the class period is a bunch of extra problems. They're not the problems you have, but a lot more problems. And I walk around the room, and the TAs walk around the room. We basically just help the students as they're working in groups on those problems, so they actually have a chance to apply the material. And at the end of the class, we go over the answers to those problems. The on-campus class also has a lab section associated with it, where they'll do crosses with fruit flies, they'll do simulations on the program Lil A1, they look at different skulls of different animals and try to identify the groups they're associated with and things like that. But it's a fun class. I mean, I enjoy it. I enjoy the online one as well. But you know, they're obviously very much intertwined.
Well, I think that's about it. We started a little late, so we went a little over time. But thank you, everybody. Sorry for the technical issues at the beginning, but thank you, everyone, for joining us.
It was a pleasure meeting all of you.
Yeah, same here. Same here.
Thanks.
Thank you.
Thank you.
I enjoyed it. Thank you.
Thank you.
Hey, but quick question: do we get extra points for having been in this?
Unfortunately, no. But thank you for asking.
All right. Well, great. We'll see you guys on the forum.
Bye.
In the other media as well. And good luck on the final and the rest of the course.
And the video, the video for this will be posted to YouTube shortly too. Thanks for participating.
Thank you.
Thank you.